Kimchi is a traditional Korean food involving various ingredients. It undergoes spontaneous fermentation, the chemical breakdown of a substance by lactic acid bacteria. Growth of these microbes can be affected by the type of vegetable, salt concentration, oxygen availability, pH, and temperature. When it comes to extending the shelf life of kimchi, it has been reported that the most important factor that affects fermentation is temperature. Ideally, fermentation occurs for either 1 to 2 days at 30 degrees, 3 to 4 days at 20 degrees, 20 to 30 days at 5 degrees to reach the optimal taste and pH levels or acidity. Over-fermentation can decrease the quality of kimchi in terms of taste and texture. A pressing problem faced by companies selling commercially packaged kimchi would be over-fermentation, thus requiring the storage at low temperatures to minimize microbial growth from packaging to consumption. However, real-world logistics vary, and accidental exposure to room temperature can occur during transportation or storage. Such incidents may last up to 48 hours, enough to resume or speed up fermentation, posing potential risks to the product quality. The impact of exposure to Singapore's temperature on the microbial community in kimchi is not well understood yet.
Hence, this study aims to simulate such an accidental exposure to assess how it influences the microbial composition, flavor, and safety of kimchi. By understanding these changes, producers can better anticipate potential issues during the distribution process, especially the safety and quality of kimchi that may have been exposed to non-ideal storage conditions. Next, we are going to introduce the experimental intervention designed to understand how the microbiome in a sealed packet of kimchi is affected by different temperature conditions, particularly a breach in the cold chain. In the control setup, we stored one pack of kimchi in the fridge at 4 Celsius degree. This simulates the ideal cold chain and fridge storage condition that is commonly used for production to consumer storage. The goal is to maintain a stable environment, typical of a proper cold chain, allowing us to compare the effects of temperature exposure. In the variable setup, we exposed another pack of kimchi to room temperature, which was about 27 Celsius degree, for around 48 hours before beginning the experiment. This simulates an accidental breach in the cold chain during the distribution or storage, where the kimchi is left at a room temperature for an extended period. This condition allows us to observe how such a breach impacts the microbiome composition compared to the control group. With this setup, we aim to analyze the microbiome differences between these two groups to determine how short-term temperature fluctuations could affect the fermentation and quality of the kimchi.
Our aim is to investigate the effects of exposure to room temperature of around 30 degrees for 48 hours on the microbiome composition of kimchi within a cold chain system. We want to understand how this temperature shift impacts the microbiome composition of kimchi. Our hypothesis is that exposing kimchi to room temperature for 48 hours will lead to increased microbiome activity. This in turn will result in significant changes to the microbiome composition, which could influence both the safety and flavor of the kimchi.
Our samples consisted of sealed 150-gram Bibigo brand packet kimchi, sourced from various supermarkets around Singapore. Batches were identified by expiry date, and a pair of packets were obtained per batch. Of this pair, one packet was placed in the control group and one in the intervention group. Control group packs were maintained in a 4 degrees Celsius environment until sampling in the lab, and intervention group packs were left unrefrigerated for 48 hours. This timing was chosen to mimic a breakdown of the cold chain over a 2-day unmanned period, typical of the widely practiced 5-day work week. After this 48-hour period, intervention group packets were once again refrigerated at 4 degrees Celsius. In the lab, kimchi packets were decanted and the liquid collected. Said liquid was sent for subsequent DNA extraction and sequencing. An olfactory and gustatory evaluation was also planned. However, due to safety and contamination concerns, the gustatory test was foregone. The olfactory test, though conducted, did not yield any noticeable difference to the untrained nose.
The first advantage of our sample collection process is minimal environmental contamination. Since the kimchi remains sealed until we open it to collect the kimchi juice samples, and by taking the samples immediately, we reduce the chances of contamination. This ensures that the microbiome composition of the kimchi is preserved and is not altered by any possible microbes in the environment. Another advantage of our sample collection is that we use 4 different batches of kimchi, purchasing a total of 8 packs with different expiry dates. By comparing these batches, we can ensure that the initial microbiome composition is consistent across all batches, minimizing any batch-to-batch variation. This control is to ensure that the difference in microbial composition between the 4 and 27 degrees Celsius kimchi group is due to the temperature difference. Studying multiple batches also reduces the risk of bias or outliers caused by handling or packaging errors. However, there might be a possible deviation as the ingredient list mentions a starter culture, but it only specified soy peptone in its starter. And soy peptone is primarily used as a nutrient source in microbial fermentation, as it contains a rich source of carbohydrates that help microorganisms grow quickly. While this might not change the type of microbes present, it could influence the overall microbiome composition of the kimchi. One disadvantage of our sampling method is that microbes localized to solid ingredients such as the napa cabbage may be underrepresented because we only collected samples from the kimchi juice. For example, if certain bacterial strains primarily thrive on the solid napa cabbage, those strains might be missed in our kimchi juice samples and lead to an incomplete understanding of the microbial diversity. To address this, we could have added a control by sampling the solid ingredients of the kimchi by blending them all together. This will allow us to compare the microbiome composition on the solid ingredient with that of the juice, and this helps to determine whether certain microbes are underrepresented.
One limitation that should be accounted for in this study was the fermentation times of the different batches of kimchi. During sample collection, the kimchi we used were from different batches. Due to this, each batch of kimchi had different fermentation times. Studies have shown that varying kimchi fermentation times led to a change in conditions such as pH levels, altering the proportion of species in the microbial population, becoming a confounding factor in measuring the effect of a cold chain break in the population. In future studies, it is still recommended to use sample pairs of kimchi of different batches, but the expiry date of each batch should be limited to within 1 week of each other. Adding on to the cons presented by Brittany, another limitation to this study was that microbes that localize to solid ingredients may be underrepresented. In future studies, it will be recommended to blend the samples to get a better representation of the species living on all constituent ingredients of kimchi.
Another limitation that should be accounted for is that the control of temperature may not have been consistent over the 3 packets of kimchi in both the refrigerated and non-refrigerated packets respectively. As we split the kimchi samples across 3 different people living in different areas in Singapore, the room temperature, humidity, and the temperature of the cool chain will not be fully consistent across all 3 settings. To mitigate this limitation, the best solution to simulate a cool chain process is to put the non-refrigerated packets of kimchi in the room temperature bath while the refrigerated packets of kimchi is placed in a chiller of 4 degrees Celsius, both running constantly for 24 hours and both equipment can be found in the lab. However, if the cost of running a bath or chiller is expensive, we could have considered only asking a member of the group to purchase the non-refrigerated kimchi and kept all the kimchi in the same condition where the different batches of kimchi will be exposed to the same environmental conditions, and another member of the group to store the refrigerated batches of kimchi in one fridge only. The last limitation to be accounted for is using our senses such as smell and taste to determine the quality of kimchi. Using smell to determine the quality of kimchi may be subjective and may not provide the extent of spoilage and the effects on the microbial population. The taste of a kimchi may indicate the changes of pH and acidity of the kimchi. However, it does not tell us subjectively what changes are present as well as any changes in microbes. Therefore, to mitigate such limitations, the experiment should be held for a longer duration with additional tests implemented together with our senses, such as changes in pH or microbes level with sequencing.